In today's session, we shall learn about citric acid cycle or Krebs cycle, which is the final common metabolic pathway for degradation of carbohydrate, protein and fat. Again, this will be a revision for you because you have all learned this in your 10+2 standard. However, acetyl CoA from breakdown of carbohydrate, fat and protein is oxidized to carbon dioxide. 2 carbons of acetyl CoA combine with 4 carbons of oxaloacetate and become citrate in the first reaction of Krebs cycle. Ultimately, in 2 steps, carbon dioxide is liberated, and in the other steps, we get oxaloacetate again. Now, since oxaloacetate is required in a small amount and acetyl CoA, a large amount, is converted continuously to carbon dioxide, oxaloacetate here can be considered as catalyst in this metabolic cycle. In the reactions, as you can see, in this reaction, actually a bond is formed between the methyl group of acetyl CoA and the carbonyl group of oxaloacetate. An intermediate is formed, citryl CoA, which is hydrolyzed to give citrate. In the next 2 reactions, as you can see, citrate is converted to isocitrate. So these 2 reactions are essentially isomerization reactions catalyzed by the same enzyme aconitase, which is non-competitively inhibited by fluoroacetate. In the next reaction, you can see isocitrate dehydrogenase converts isocitrate to oxalosuccinate. NAD is converted to NADH. This NAD is specific for mitochondria. There are other NADs which can be found in cytoplasm. The next reaction is spontaneous, catalyzed by the same enzyme isocitrate dehydrogenase, and where carbon dioxide is liberated. This reaction is a very important reaction. This is an example of oxidative decarboxylation, where alpha ketoglutarate is converted to succinyl CoA with the help of alpha ketoglutarate dehydrogenase. Coenzyme A is required along with some other cofactors. If you remember, I had told you the same cofactors which are required for the pyruvate dehydrogenase enzyme complex, that is, along with coenzyme A, NAD, FAD, thiamine and lipoic acid. So 5 cofactors in total are required for this enzyme complex and also PDH. This reaction is irreversible and is inhibited by arsenite. Carbon dioxide is liberated. The next reaction, catalyzed by succinate thiokinase, converts succinyl-CoA to succinate. Coenzyme A is liberated. ADP is converted to ATP. This energy, that is conversion of ADP to ATP, comes from actually this reaction. In other words, this reaction liberates so much energy that not only NAD is converted to NADH, this NADH to ATP, but succinyl-CoA which is formed when it is converted to succinate can convert ADP to ATP. So the energy of this reaction converts NAD to NADH and ADP to ATP. Importantly, this reaction is the only example of substrate level phosphorylation in Krebs cycle. In the next reaction, succinate dehydrogenase converts succinate to fumarate, but this is the only reaction in Krebs cycle where FAD is converted to FADH2. In other reactions, either NAD is converted to NADH or ADP is converted to ATP. This occurs, that is, FAD is converted to FADH2 because the energy of conversion of succinate is not sufficient to convert NAD to NADH. You know, in the old convention, from one NADH we get 3 ATP in the respiratory chain by oxidation. From one FADH2 in the respiratory chain we get 2 ATP. So by this I meant less energy is produced in this reaction than this or this reaction. Where 3 ATPs can be formed, here 2 ATPs can be formed. This enzyme has iron sulfur protein, and you might have learned in enzymes that this enzyme is inhibited competitively by malonate. So malonate inhibits this enzyme. In the next reaction, fumarate is converted to L-malate by fumarase. Fumarase is specific for L-malate. In the last reaction, L-malate is converted to oxaloacetate by malate dehydrogenase. NAD is converted to NADH. Now, this reaction is reversible, and if this occurred outside our body, the equilibrium of this reaction would favor the conversion of malate from oxaloacetate, not malate to oxaloacetate. But in our body, malate is always converted to oxaloacetate. Why? Because the product of this reaction, oxaloacetate, is continuously condensed with acetyl-CoA and removed as citrate. So since the product becomes less in concentration, the equilibrium of this reaction always goes in this way. Another thing, in this cycle, no oxygen as such is observed. There is no oxygen directly involved. But Krebs cycle is aerobic, essentially aerobic. Why or how? because the NADH or FADH2 must go to the respiratory chain for oxidation to regenerate FAD or NAD. Otherwise, this cycle would stop. So, unless FADH2 or NADH is oxidized in the respiratory chain with the help of oxygen, NAD or FAD will not be regenerated to continue the cycle. So, Krebs cycle is linked very much to the respiratory chain and therefore is indirectly aerobic. Energetics of Krebs cycle. As you can see, 3 NADH from the reactions catalyzed by ICD, alpha KGDH and MDH, 3 NADH formed, which in the respiratory chain are converted to 3 ATP each, that is 3 into 3, total 9 ATP. From 1 FADH2 in this reaction we get 2 ATPs in the respiratory chain, and in this reaction we get 1 GTP or ATP. So in total, by one turn of Krebs cycle we get 12 ATPs. Krebs cycle is both amphibolic and anaplerotic. The word amphibolic means like amphibians, that is, it has two opposite roles. So, an amphibian, a frog, can live both on land and in water. Similarly, Krebs cycle has both anabolic and catabolic roles. As you can see in this diagram, from citrate, fatty acid can be synthesized. From alpha ketoglutarate, glutamate can be synthesized, and so on. So these are the anabolic roles of Krebs cycle. On the other hand, from amino acids like methionine, valine, isoleucine, succinyl-CoA can be formed. From tyrosine, fumarate can be formed. So these are the catabolic roles where these amino acids are catabolized through Krebs cycle. So Krebs cycle has both anabolic and catabolic roles and therefore is amphibolic. Now, by anaplerotic role of Krebs cycle, I mean that when an intermediate of Krebs cycle is removed, suppose succinyl-CoA is removed as heme. So succinyl-CoA is a starting material for synthesis of heme. This gap is filled by something else, like some amino acid. So whenever an intermediate of Krebs cycle is removed for some purpose, that gap is filled by something else. So this is the anaplerotic role of Krebs cycle. So I am not going into details. You should remember this flow diagram. Regulation of Krebs cycle. Firstly, the availability of ADP and therefore NAD. So the sites of regulation are the reactions catalyzed by citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and you may say pyruvate dehydrogenase. In turn, these enzymes are stimulated by calcium, and individually citrate synthase is inhibited by the ultimate products of Krebs cycle like ATP, NADH. Isocitrate dehydrogenase is inhibited by the same products ATP and NADH and stimulated by ADP. Alpha ketoglutarate dehydrogenase inhibited by NADH and succinyl CoA, the product, pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase both are inhibited by ATP. With this, I finish Krebs cycle. Thank you.